and she received recently last year the IBRO Earlier Care Award for this particular work. So I would like to acknowledge IBRO as well as acknowledge the efforts of Dr. Ayushi Parikh. And secondly, I would like to acknowledge my university. You can see this is a lush green campus. It's the aerial view of my university. It's the largest women residential university in the world. It is spread across 1,000-acre campus. So you can see some, good research is going on in this campus with this lush green campus. So I would like to conclude my talk with the great saying of Mahatma Gandhi, father of nation of India, about my university, Vanasthali Vidyapith. I'm very anxious to visit Vanasthali, and Vanasthali enshrined in my heart. So we follow Mahatma Gandhi ideology. That's why I would like to quote Mahatma Gandhi's for our university. Thank you so much.
That's all. That's all.
It was a really wonderful, thought-provoking lecture, and you explored on the specialized and advanced topic in the field of pharmaceutical sciences, particularly in the era of nanotechnology and drug delivery systems, and key aspects and takeaway on this lecture. So you make it very interesting. So very innovative approach you presented relevance to neurodegenerative diseases, and this is a interdisciplinary approach you provided. And topic combines knowledge from the pharmaceutical sciences, nanotechnology, neurosciences, making it engaging subject for the academic audience. Potential impact also research could be lead to development of new treatment and therapeutic strategies for Alzheimer's disease, benefiting patients and the healthcare sector. Overall, this lecture was likely informative, engaging, and sparked interest in the audience, given its focus on cutting-edge research and potential therapeutic applications. I'm really very happy to chair this session, and I know Ashutosh from many years. He is a very dedicated and wonderful scientist. His work is referred by all over the world. He's getting so many accolades and awards. So I wish you all the best again in future. And I also salute his better half, Ayushi, that she's also providing impact in this combo research, you can say. So thank you so much, and really wonderful. We are very blessed to this lecture. I request the audience, if you have any questions, please you can ask.
Trials in human being, and it is EVR loaded is approved by the drug controller of India, or is still in the animals only?
Sir, so far we have studied the bioactive as well as the formulation loaded with that bioactive substance at a preclinical level only. However, since it is a very beginning, but it has a potential of translating to human as well. So we are under scale, sir. But currently, if you ask, it is at a preclinical level
only. Sir, whenever we are talking about the route of administration, so during your animal study, what basic problem you have faced? Because the nasal cavity of animal is so much less, and even oral delivery is so much difficult in the
very potent concentration, and we administered somewhere 50 microliters at a time to the one side of nostrils, and followed, kept it for 5 to 10 minutes, and then we administered to the second side. So this is how we manage the effective loaded concentration to the nostrils of the rat. This is how we proceeded. This is the biggest challenge my research scholar usually face, because some of them are working on PD and they are using mice. So at the same time, this issue comes over there as well. But the most important thing is when you want to administer drug intranasally, you need to keep the drug concentration at a higher level. So that's why we have prepared a suspension. And we administered in a periodic manner, 5 minutes interval.
Because whenever we are using high amount of drugs, that time some animals may get allergy during that time, and that may be the basic problem of the basic issue.
Very rightly said. So I haven't showed here, but we performed a serial toxicity study as well. One important point when we talk about nasal delivery system, the biggest challenge that we face, there used to be a chances of getting irritation into the nasal mucosa. So for that we studied serial toxicity study as well for 24 days. We keep administering drug for 24 days, and after the end of the 24 days, we observed if there any kind of erosions or abrasions is there in that mucous membrane of the nostrils.
One more question. Which formulation will be most suitable for nasal delivery?
Very tough question, but yeah, I would suggest the biggest issue with the nasal cavity is you need to keep the drug into the nostrils for a longer duration of time. At the same time, because there used to be a ciliary excretion, clearance is there. So if you want to select any nano formulation, you have to keep in mind that drug needs to be permeate at a faster rate from that particular reason. At the same time, you have to keep some kind of mucoadhesive substance there, so that will increase the residence time of that formulation as well as the drug to the nasal mucosa.
And one more problem during the formulation, actually whenever we are trying to go for novel formulation, just nanoparticle and something else, that time entrapment efficiency and loading that will also make some problem during if we want to give animal to high dose, because the entrapment efficiency and loading efficiency also hinder that thing.
So we are fortunate enough in that case because we have entrapment efficiency more than 90% and loading was somewhere 19%. So yeah, absolutely rightly mentioned, but that you have to decide. That is how you have to proceed with in what particular scenario, which combination of excipient will be suitable, which will increase the loading as well as entrapment of that particular drug. That you have to assess and study.
Thank you very much, sir.
So behavior results always comes out right, sir?
Pardon?
So all behavior results always comes out right, sir?
You want to say that are they trustworthy?
Yes, sir.
This is your direct question?
Yes, sir.
Okay, so absolutely right. If you see that standard deviation, if you have seen in my graph, there is a lots of variation. Even in a days to day, if you compare, there used to be a lots of variation. 10 to 20% variation used to be there. So the best way to proceed, in my opinion, you cannot alone rely on the behavior analysis only. But at the same time, when you choosing the group of animals, you need to keep as fair as possible, so that you will be getting ultimate at the end, standard deviation, standard errors. If you are doing in a right way, that will help you a lot. And at the same time, that analytical technique, statistical technique you are using, that needs to be robust enough to identify the variation, whether it is significant or not. That is very important, as far as my understanding is concerned.
Congratulations, Dr. Parekh, for such a nice work. One question which is arising in my mind is that you have done brain uptake study and pharmacokinetic as well. And you have compared oral as well as intranasal delivery of the drug. And the question is how you meter the intranasal dose. And by orally you will be giving a fixed dose. And by intranasal route, how it is possible to give the similar dose which is given through the oral?
Ma'am, I think you want to ask me whether we kept the drug loaded orally as well as nasally, whether it was equivalent, same or not. That is your question, and how we manage. So, ma'am, it was same. What we did, oral dosing or nasal dosing. Generally, when you are comparing, there used to be a ratio of 1/10 you can cap particularly, because there are two different scenarios there. If we talk about oral administration, there used to be a long residence time if we compare with the nasal route as well. So how we kept similar, if it is your query, then we calculated equivalent concentration of drug, and as I mentioned to her, we administered 50 microliter. It will vary. Sometimes it requires 35 microliter, 40 microliter, depending on the concentration we are able to load in that particular formulation, which is again depends upon the entrapment efficiency and loading of the drug to the particular formulation. So while keeping that entrapment in mind, we varied the loaded dose. There was 8 to 9 formulations, so we administered different volume of drug to equalize the amount of drug administered nasally as well as.
Yes, sir.
Yes, sir.
When we are using this intranasal route, is there a chance that the drug goes to the lungs and create problem? Because even if we are using oral dose itself, it can go to lungs, right?
Lungs.
Yeah, lungs.
Yeah, absolutely. So what is the question?
Is that a problem? Like you need to make sure that the drug stays in the nasal cavity, not to the lungs, right?
Yes. So for that, systemic absorption you've seen, if we compare with the oral, it was quite less, right? So with the fraction of drug reaching to the blood is less, then the chances of reaching to lungs is again quite lesser. But since you asked me how we managed, so basically this SLN we loaded, that was instilled and admixed earlier in some mucoadhesive polymer. So they have a tendency to adhere to the mucosa of the nasal. So which cap them to that localized area of the nostrils only. So that is how we manage that. We will rule out from the excretion as well. So that is how we proceeded.
Thanks for the wonderful talk. How you manage sneeze during intranasal administration? Do you use antihistamines?
No. We haven't administered any antihistamines. All grade goes to my research team. But yeah, how we manage, if you give sufficient time and if you handle in a proper manner, and as I mentioned, that nozzle of the micropipette from which you are administering, if it is giving a proper space and if you are administering very slowly, there is a very less chances of having any sneeze. Sometimes it may happen. I'm not showing absolutely 100% we were sure, but in that case, what we used to do, we escape that dosing. At that day, we usually not given any particular further dose. We will wait for the next day only.
Thank
Thank you so much. With our speaker is so enthusiastic, as well as our all audiences also. There are so many questions and you give wonderful answers. I'm very much Vivek sir for inviting us in this international conference and this chair this session. As well as I'm very much thankful to Julie ma'am, Sunita ma'am, as well as Garima and Lalit sir for inviting us. Thank you very much, sir.
Thank you so much, sir. Thank you so much. Thank you, Dr. Vivek, sir, for inviting me again here. So this is a really nice experience I have here. Thank you so much.
Thank you, sir, for your enriching lecture. Now, I will request Dr. Garima Joshi and Dr. Vivek Jain to felicitate our speaker, Professor Ashutosh Parekh. I would also request them to felicitate our chairperson and co-chairperson.
Thank you, sir. It is my privilege to invite Dr. Sandeep Kumar Singh from AIMS Nagpur to deliver his lecture on the power of Drosophila model in Alzheimer's research, a platform for novel therapeutic drug targets and discoveries. With a great respect, we invite our chairperson, Dr. Komal Sharma, Professor of Pharmacology, Bhopal Global Institute of Pharmaceutical Sciences, Udaipur, and the co-chairperson, Dr. Meenakshi Bhargatia, Associate Professor of Pharmaceutics, Bhopal Global Institute of Pharmaceutical Sciences, Udaipur, to take their seats. To welcome our speaker, I would like to call Dr. Sunita Panjawar to do honors. Now I will hand over the mic to our chairperson and co-chairperson.
Hello. Good afternoon, everyone. I am Dr. Meenakshi Bhargatia, Associate Professor, DM Institute of Pharmaceutical Sciences, DM University. It's my pleasure to welcome you all once again to this technical session of the International Conference on Recent Advances in Neurochemistry and Neurological Disorders. It gives me great pleasure to introduce our distinguished speaker, Dr. Sandeep Kumar Singh, currently working as an assistant professor in All India Institute of Medical Sciences, Nagpur. He is a well-trained neuroscientist with a strong background in biochemistry and biomedicine. He earned his PhD degree in biotechnology from the most prestigious institution, the capital of knowledge, Banaras Hindu University, Varanasi. His research interest includes neuroscience, molecular biology, neurodegenerative diseases, aging, nutraceuticals, and nanomedicines. He has more than 12 years of experience in neuroscience research. He has used animal model systems to investigate the therapeutic potential for Alzheimer's disease using synthetic as well as natural compounds in Drosophila as a model system. Dr. Singh has published more than 65 articles in Journal of International Repute, such as Journal of Alzheimer's Disease, Pharmacological Research, Current Neuropharmacology, and many more. He has been also credited with 12 book chapters and editors of 5 books. He is also member of editorial boards of several international journals related to the field of neuroscience. He served as a guest editor and handled a special issue in various international journals related to the field of neuroscience from MDPI, Frontiers, FBL, etc. He is also the member of several scientific societies like Indian Academy of Neurosciences, Alzheimer's and Related Disorders, Society of India, American Academy of Neurology, International Society of Neurochemistry. Bentham Science Publisher has also appointed Dr. Singh as a Bentham Ambassador. He has attended several scientific national and international conferences in India and abroad and presented his research work. The title of his presentation is The Power of Drosophila Model in Alzheimer's Research, a platform for novel therapeutic drug targets and discoveries. Now I invite Dr. Sandeep Kumar to deliver his talk.
Thank you so much for the kind introduction, ma'am. first of all, I would like to thank Dr. Jansen who has invited me for this wonderful conference and where I can present my work. So, as in past, several speakers have presented their wonderful research data and their discoveries and their therapeutic implications. So, today my presentation is basically focused on how we can use the Drosophila as an AD model. It's not about the data and all. I'm just presenting the things here. How you can create the Drosophila AD model as per your desired gene, which you are going to use in your research and in your study. So, I have divided my talk in two parts. The first will cover the Alzheimer's disease, its therapeutic implications, treatment options, and the second part will cover the Drosophila model system used in the AD research. How you can create the Drosophila model system depend on your study gene like MLI beta, tau protein, or whatever you want to study. You can prepare your gene, your strain accordingly. And what are the different mechanisms you can use in the Drosophila model system and different pathways you can study. So, and phenotypically, how you can express your protein in particular region like eye tissue or neuronal tissue. So that I will deal about. So basically, this will be important for the student who are planning to involve the Drosophila model in their AD research. And so let me introduce first. I will talk about little bit about Alzheimer's disease. As already all the speakers have spoken about the what is Alzheimer's disease and how it is originated, what are the statistics and all. So I'm quickly going through these points. So as we know, the Alzheimer's disease is basically the irreversible neurodegeneration of the brain, and in this, eventually there is a complete deterioration of the brain, basically in the hippocampus and the cortex region. So in present scenario, there is estimation about 7.2 million Americans are aged 65 and older are having Alzheimer's disease. So the expectation about 2050 is about 15 million people will get affected by the Alzheimer's disease. So worldwide, approximately 57 million people had dementia in 2021 statistics with Alzheimer's, and it contributes about 60 to 70% cases of dementia. So as we know, it is the most common cause of dementia among people who are age 65 and older. Alzheimer's is the sixth leading cause of the death in the United States of America. Now, in context of Indian scenario, there are approximately 8.8 million Indians over age of 60 and above are currently suffered with the dementia. It was earlier it was 3.8. So you can see how dramatically increasing the numbers of people affecting with the Alzheimer's disease in India. So recently, the studies from the India and Africa they showed that the main affected people are from urban area in comparison to the rural area. And what is the reason behind this? This could be what is the deciding factor? It could be a life expectancy. Had already Dr. Mandal has told that nowadays the life expectancy increase in India. So this could be one reason. And the second is it lifestyle? Yes, of course, it is also one reason. And there is diet habit. It is also one reason for increasing incidence of Alzheimer's disease. So these are some common symptoms of the Alzheimer's disease. I'm not going through one by one. You just telling you in a quick moment like memory loss, inability to perform daily activities, mood and personality changes, psychiatric symptoms. And the severe case, the person is lost all the complete memory, and even one must need a caregiver to take care of them in severe state. So these are the stages of the Alzheimer's disease. So in normal stage, it is a forgetfulness stage when the Alzheimer's start. So this is a normal complaint. People are just forgetting things, daily things. The second is the mild cognitive impairment, where the person used to go and perform their work, but their work capacity is little bit hampered. And the mild Alzheimer's stage, the most common function deficit in these patients. Basically, they used to perform. They are unable to perform daily activities like they used to cook the food, and they forget to like put some salt or some anything like so. This kind of activities, this is comes under mild Alzheimer's disease. What happens in the moderate Alzheimer's disease? And this is the very manifestation where is the decrease in the ability to choose proper clothing, depending on the weather condition. Even one can forget what to wear or what to not. So in the moderately severe conditions, at this stage, the ability to perform basic activities is completely hampered. And the last and the severe Alzheimer's stage, this is the complete, you can say a gone kind of state where person should need 24-hour caregivers. Without that caregivers, one cannot survive. So these are some basics, causal factors of Alzheimer's disease. As you can see, the E's factors are interlinked because, as we know, Alzheimer's disease is a very complex disorder. This is not caused by a single element. So there is nutrition depletion, heavy metal toxicity, mitochondrial destruction, oxidative damage. But these are interlinked. If you are targeting one thing, then you cannot find a drug. That's why people are facing challenges to discover any particular drug for Alzheimer's disease because they are unable to target all the complex things in a one target approach. So that is the reason some people are focusing on nutrient depletion, some people are focusing on heavy metal toxicity, some people are focusing on mitochondrial dysfunction. So there are different causal factors of Alzheimer's disease. When we talk about the pathogenesis, so there are different pathways, different cascade people are reported. Just one is very famous is amyloid hypothesis. As many speakers have already spoke about this amyloid hypothesis, so I'm not going into the detail. And there is tau hypothesis, inflammatory hypothesis, microbiota gut-brain axis hypothesis, abnormal autophagy hypothesis, metal ion toxicity, and cholinergic hypothesis, glutamate excitotoxicity. So these all are included in the pathophysiology of the Alzheimer's disease. So among these two are basic focus to find out the drug targets: amyloid hypothesis and tau protein hypothesis. But people are nowadays focusing on the gut microbiota hypothesis also. So I will start with the amyloid beta because this is the main hallmarks of the Alzheimer's disease. Amyloid beta and tau protein. These two are the main hallmarks of Alzheimer's disease. So how Alzheimer's disease is amyloid beta protein generated basically. So it is what is amyloid beta protein? First, it is 36 to 43 amino acids long peptide, which is generated by the ATP processing inside the cell. And the prevalent peptides are among these 36 to 43 are 40 and 42. Among 40 and 42, 42 is the most toxic fragment, which is actually accumulate and cause the senile formation, which results in the degeneration of the neurons, hippocampus area neurons, and damage of the reticular neurons. So how this cascade happens actually? There is a generation of monomers after the ATP processing, and then these monomers, soluble monomers, will combine and form the insoluble fibers, which is called amyloid plaques, and which leads to the Alzheimer's disease. And why this happens? Because there is in body there is a clearing mechanism also there. So if there is imbalance between the production of these amyloid peptides and the clearance of these amyloid peptides, then accumulation of this amyloid beta happens. And why this happens? Because when there is APP, next slide I'm just explaining. When there is a processing of APP and there is a beta secretase and gamma secretase are active, then there is generation of amyloid beta. So in older age, what happened actually, the activity of beta secretase and the gamma secretase increased. That's why the production of the amyloid beta increased and the clearance mechanism is slowed down. So that they start the accumulation of the amyloid beta, which cause the degeneration of surrounding neurons in the hippocampus area. Otherwise, APP is present in every human and every cells starting from the birth and age people, but they are effectively working when the people are going older because the activity of this beta secretase is increased and ultimately result to the amyloid beta. This amyloidogenic pathway, the activity increase in the older one, and this is the normal non-amyloidogenic pathway because the SAPP alpha is required for the normal functioning of the neurons. So that's why the processing of this alpha secretase and gamma secretase is normal for normal function, But when there is a beta secretase and gamma secretase activity increases, then there is generation of A beta, which is neurotoxic, as you know. So, what are the characteristics of this amyloid beta protein? Basically, it is insoluble and it deposits in the brain. Main component, I already told you, 40 and 42 amino acid long. It is found in the hippocampus and the cerebral cortex area. It is toxic and disrupts the synapses and neuronal function. It activates the immune response also and triggers neuroinflammation and oxidative stress. Amyloid beta is more aggregation prone to toxic than beta 42, I already told you. And this is also associated with the mitochondrial dysfunction. So second hallmark is the tau protein. So what happens in case of tau protein? Tau protein is one thing I forgot to tell you, that amyloid beta is always present extracellularly and the tau protein are always present intracellularly, because the deposition of the amyloid beta is happen outside of the cell, and the intracellular deposition of the hyperphosphorylated tau is happen inside the cell. So in tau hyperphosphorylation, what happens in normal neurons? You can see in healthy neurons there is a microtubules which are intact by this tau protein. You can say that tau protein is stabilizing these microtubules by keeping them intact, which is required for normal function. But in diseased condition, when there is a hyperphosphorylation of tau happens, then this start disintegration of this microtubule, and there is a formation of the tangents, which you can call neurofibril tangents, which is results into the formation of the clumps outside the neurons and start the degeneration of the neuron. So now come to the therapeutic aspect of the Alzheimer's disease. There are several therapeutic aspects people are exploring nowadays, like non-pharmacological interventions like physical activities, musical therapy, social activities. There are several FDA-approved drugs which are based on cholinergistic agent like memantine, and this aducanumab is antibody-based drug, which is based on the amyloid beta antibody. People are also using alternative approaches like vitamin E, melatonin, calcium, and also people are using herbs as an alternative treatment like ginkgo biloba, saffron. People have reported their neuroproductive activity in different literatures. So, but all in cumulative, the therapeutic approaches, all the therapeutic approaches combined to try to resolve the issue which is related to the cognitive function, behavior, memory, attention, reducing oxidative stress, neural plasticity, and neurogenesis. So, as far as the treatment concerned, there is no any treatment for Alzheimer's disease available in the market. There are several clinical trials already been happened. Several clinical trials have been failed. Recently, this donanemab, which is just in past the third phase of clinical trial by the Eli in July 24, and it has shown about 35% of reduction of the amyloid beta burden in the brain. But again, these all drugs are symptomatic drugs. There is no 100% efficacy, and there is also side effects. So other people are also trying to find out the anti-tau therapies that are in earlier stage, and gene therapy with focus on the neuronal growth factor. Also, people are trying, and so this is the continuous journey. People are trying to find out the treatment of Alzheimer's disease anyhow, but till now there is no success in complete therapeutic approach or complete drug. So these are the, you can say, journey for the drugs approval from 1993 to 2023. These are some cholinergic agents. These are the antibody-based drugs. So this is the kind of pathway from the year-wise we are showing here. So now second part will come to the Drosophila model system. So as we know, people are using different model systems to study the Alzheimer's disease on the molecular basis. So why Drosophila model? This is a very fantastic model and my favorite model. Actually, I worked on Drosophila. So why I have chosen Drosophila and why this is a very important model? Because the first point, this is having very short generation time. Its life cycle is about 10 to 12 days. So you can generate very fast. Relatively genetic manipulation, there you can do a lot of genetic manipulation. You can play anyhow with the Drosophila. You can manipulate any gene. You can insert or delete or mutate any kind of gene in the Drosophila. This you cannot do in the mice easily. This is very costly and very tedious one. And they have very low chromosome number, only 4 chromosome is there in the Drosophila, and they have a lot of molecular conserve mechanism also. They have shown the homology with the human genome also and functional orthologs. And the most important thing is don't need to ethical approval from any committee. If you are working with Drosophila, you don't need to ethical approval from any committee because this is invertebrate model and this is not comes under this ISUC guidelines. So you can start anytime working with Drosophila. You don't need to wait for ethical approval and all. So that's why this is the best model. So this slide is showing the life cycle of the Drosophila. As you can see, you can count the days, about 10 or 12 days. When the fly will mate, they will hatch out the embryo in the food, and after first star larvae, then second star larvae, the third star larvae, and it will turn into the pupa in 2 to 3 days. After pupa, around 3 to 4 days, it will turn into the adult. So in 10 days, your animal model will be ready to do the experiment and all. So I'm showing here I imagine this are 13 star larvae because in future slide I will show why this is important and how you can check your particular protein expression in these eyes. Phenotypically, you can see the changes, the toxicity by the any protein like amyloid beta or tau protein. This is the adult eye, how beautiful it looks like. When you express the protein in eye tissue like amyloid beta, this structure got distorted. It will show clearly phenotypically. You can see, you can visualize your drug effect on your normal food treatment. So here I'm showing the amyloid beta modeling in Drosophila, but you can do the modeling by using all these aspects like amyloid beta models, tau models, APP models, or you can use your dual model to see the severity. If you combine co-express the amyloid beta gene and tau gene in single model, then it will show higher propensity and higher toxicity in compared to single model. So basically, amyloid beta is a very common and very prone, so people are using this amyloid beta protein insertion inside the Drosophila. So I will discuss in the future slide the amyloid beta model only, not all. So the amyloid beta modeling in Drosophila, how we will do this? Actually, as we know, the amyloid beta fragment is the main toxic fragment in the Alzheimer's, which is generated by the APP processing. But in Drosophila, there is APP is not present. Instead of APP, there is present of APP-like gene in Drosophila, which itself cannot generate the A-beta-42 fragment. So that's why what we do actually, we just insert the human A-beta-42 in the genome of the Drosophila, and that transgene will be used as a model. We used to express that particular protein, and that is having human amyloid beta, not Drosophila. So you can clearly, whatever compounds or drugs you are screening on that strain, you can say that strain is effectively working on amyloid beta 42. So that's why in that way we can generate the strain of the amyloid beta, and which will express the amyloid beta toxicity, intracellular expression of the human amyloid beta fragment. So in transgenic Drosophila model, A-beta-42 forms cytoplasmic B amyloid, beta amyloid with the properties similar to those found in the human brains, because you are expressing human amyloid, not Drosophila. So how you will do that? How you will express your amyloid beta-42 in Drosophila? You will take different strain. Have you heard about the US Galford system? Anybody? This is a gene expression system which people are using to express any kind of gene in tissue-specific manner. Suppose we have a human amyloid beta gene and we would like to check their expression in a particular tissue, like in eye tissue or in neuronal tissue. Then what we'll do? We'll just take the neuronal or eye-specific GAL4, which is you can say enhancer, a driver, and we put our gene under US control, upstream activator site control. Well, we'll insert, this is human gene is amyloid beta protein, amyloid beta 42, and we'll insert this gene in Drosophila under US promoter. So these gene will not activate until and unless when GAL4 will go and bind with this activator, and then activation or transcription of this gene will happen. So you are taking US Gal4 line, which is having human amyloid beta insertion, and if you want to express in IGAL4, then what you will use IGAL4 lines. Otherwise, if you would like to express in neuronal to check the motor activity and other behavior, let's say in Drosophila, you need to express these protein in USGAL4 using ELAG-GAL4. So in next slide, I will show how you will set the process by using these cell lines, these strains of Drosophila. There are some transgenic lines like A-beta transgenic line. This is the genome of the transgene US-A-beta /CYO tube gal80 TS. So this is the transgenic line which the Bloomington Stock Center has created, and this having the human A-beta 42 insertion in their genome. So these fly itself will not show the expression of A-beta 42 until and unless you will not make a cross with other Gal4 line. Suppose you would like to check the expression of this amyloid beta 42 in the eye tissue, then you need to fix a cross between IGAL4 and USGAL4. I will show in next slide how you can set a cross and what kind of progeny you will get in the F1 generation and how you can select on the basis of their balancer. So I will speak in the next slide. This is a kind of vials where you can culture the Drosophila. Anyone here is working on Drosophila or not? Nobody is working on Drosophila. Okay. So this is the kind of culture medium where you can rear your Drosophila fly, and this is the fly which is a non-curly wing, and this is the curly wing. The significance of curly and non-curly I will explain in the next slide. This is the cross which you need to set if you want to see the expression of that particular protein which you have inserted under US promoter. Like you will take this line US A-beta 42 and IGAL4. So if you will set the cross, Mendelian genetics, you all know how you are going to produce the F1 generation. What are the combinations you will get in the F1 generation? This is what showing here. So you can see in this combination, there is US A-beta and Gal4 come together. So that's why the expression of amyloid beta 42 protein will happen in this combination only, not in these three. So how you will select it? Because the F1 progeny will be in the single vial where all the flies. So how you will select that these our express protein is present in this fly only. So that is, there is again a beautiful thing in the Drosophila. The balancer are there in different chromosomes. So the cardiobalancer are present in the second chromosome of the Drosophila. So what happened? If you can see here, the US A-beta 42, IGAL4, there is no cardiobalancer, so these fly will be non-curly. But in other three, recombination, you can see there is a cardiobalancer. So the fly which is having curly wing, they are not having our desired gene in their genome. So we just select out on phenotypically on the basis of their curly wing and non-curly wing. So now these fly which is having a non-curly wing will express the amyloid beta protein in their genome, and these are our experimental strain. So if you will expressing in the eye tissue, then what happen next? Their amyloid beta toxicity will be in the eye. Here you can see the normal eye. It's a wild type eye. The fly which is Oregon R fly, you can say a wild type fly, which is having normal material with the clear bristles and all. But when you have driven your fly in a normal food which is having amyloid beta insertion in the genome, then you can see clearly there is a reduction in the eye size, there is disruption of the ommatidia, and there is a lot of deformities in the eye happens. Same you can see in the scanning electron microscopic images. Here the ommatidia is intact, bristles and ommatidia is clearly visible, but in the A-beta fly which you have driven in the normal food, they are showing their disruption in the eye, and there is a lot of damage in the eye tissue along with the bristle loss. So this kind of phenotypically characterization you can see, and if you are administrating with some drug and you can see there is improvement in the eye degeneration. So in that way, phenotypically you can see clearly the effect of your drug or treatment, whatever you are giving in your. So this is a very good model for screening large number of compounds if you are doing in a very short time. So this model can be used. So this is again immunostaining of third star larvae. Imagine this. So we can see the green spot are showing the strain, amyloid beta strain, and the red is actin antibody staining, and this is nuclear stain. So the green dots are showing the amyloid beta aggregation is third star larvae, eye imaginary tissue of the third star larvae, which confirm there is a lot of aggregation and lot of deposits of the amyloid beta present in the eye imaginary disc of Drosophila. So when you will supplement it with some drug or some compound, then you can compare your results whether they are down regulating or up regulating or not. So that is another aspect of the research. In context of behavioral assay, you can do a lot of experiment like climbing assay, memory assay, olfactory assay, social interaction assay in Drosophila also, like in mice and other model system. So just the climbing assay I'm telling about here. What happened in the climbing assay? This assay is basically used to check the motor activity of the Drosophila. So for this assay, you need to express your this amyloid beta protein in neuronal tissue. Then only you can check whether this motor behavior is happening in the Drosophila or not. So what we need to do in this experiment, you just need to, as you know, the flies are the negatively geotaxic. When you will tap in the wire, they will start move up. But when they have the human amyloid beta insertion in your genome and there is amyloid beta toxicity, so they will unable to move in compared to the wild type fly or treated fly. I will show you this video so you will understand in what way this experiment can be carried out. So what are the parameters? You need to measure the climbing speed. If there is deformities in the neurons of the Drosophila, they are unable to climb up as compared to the wild type fly or the fly which are not expressing the amyloid beta protein. Their success rate and their distance of climbing. So this kind of activity is showing in this chart. You can see you need to monitor from the day 5 to day 10, and you need to take 10 to 20 flies in every vial, and repetition can be done in the 6 vials and 3 times every day. So number of fly climbing here, the percentage, and here is the number of day of post closure. So you can see the green bar is showing the 80 flies in the normal food. That means they are expressing the amyloid beta protein in their genome, and they will show the less climbing ability as compared to the flies reared in the different concentration of foods, or you can say controlled fly. These are the controlled fly. They can climb up 100%. I mean, if you tap, then all the flies will almost able to climb up to the vial. But in the fly which is having expressing the amyloid beta 42, they are not able to climb up to the top of the vial. So can you play this video, please? Just double click on. उस पे क्लिक करो वीडियो पे. This flames. You can click on this only. You can click on this. I have set a video here. So anyways, this is not playing. I don't know what is the happen. Actually, you can see in these are the different vials having amyloid beta 42 flies, 11 controlled flies, and different treatment flies. So when you tap these flies, they will start to climb up into the vial. In controlled fly, you can see all the flies are able to climb up to the vials, and the treated and the flies which is having amyloid beta 42, they will not able to climb up. Otherwise, if they start climbing up, they will fall it down. And again, they start to climb and they are not able to reach on the top of the wire. So this is the actually assay to check the behavioral aspect of the Drosophila locomotor behavior, you can say. The second one is longevity assay. This is also one experimental assay by which you can measure the longevity of the fly or lifespan of the fly, because the fly which having human amyloid beta insertion in their genome, they cannot live for long. They will die early age. So to measure the complete, death of all the flies in your vial, you need to measure alternate day, and you are counting the dead fly each and every day, and you will perform these experiments until all the fly will dead. So in this graph, you will see that the fly which is having the amyloid beta insertion in their genome, they will have life expectancy around 42 or 43 days. But the fly which is of normal control, they are having about 70 to 75 days of life expectancy. In the same way, when you are treating with some compounds, then it will just enhance the life expectancy as compared to the fly expressing the amyloid beta. So it will show that there is a toxicity due to the amyloid beta, which reduce the life expectancy in the Drosophila as compared to the fly, which is in normal control. So there are some pathways which you can explore in Drosophila, like amyloid beta toxicity related pathway, DNK pathway, MAPK pathways, and which leads to the neurodegeneration. These pathways you can explore in Drosophila also in the same way, in a very fast and accurate way. The tau activities you can check by using this Drosophila model system. Neuroinflammation activity you can check, like how immune system responses against any drug or any compound you are giving them. And autophagy and proteostasis you can study, and oxidative stress parameters related damage you can also check in the Drosophila model, like increased ROS and mitochondrial dysfunction, and all the aspects which you are related to AD, which you can check in other model system, you can easily implement in the Drosophila model system also. So you can also implement the personalized medicine and gene variability in the Drosophila model, because they have a lot of genetic background, and you can play with different combinations in the Drosophila, which is not easy in mice model or rat model, because they have different genetic background, which is related to the AD genes. The modeling human gene variants, you can generate a different mutation using ApoE gene, PSE1, amyloid beta, tau, any kind of mutations you can generate, and genetic variation you can create. And there is genome-wide screening, identify genetic modifiers of AD-related phenotype. This is also one important thing by which Drosophila is important for AD research. The testing personalized treatment, Drosophila model test treatment tailored to specific genetic background are variants. So gene environmental interaction, you can also test any diet or any stress effect in the Drosophila model, and how influence this model is about to AD risk. That is why again in the AD model, like in amyloid beta, tau, you can check. Inside into genetic risk factor, Drosophila model is informed human genetic studies and identifying potential therapeutic target, because in all the disease you can use the Drosophila model, because there is homology with the human like 60 to 70%. So in any kind of disease you can use, not in only AD. So what are the future perspective of the use of Drosophila AD model in research? You can do the genome editing, because there are a lot of recombination you can generate in the Drosophila using different genetic aspect, genetic background, and integration with the human data sets. Because already I told you there is a homology between the human and Drosophila, so you can integrate that data with the human data sets also. So basically, behavior assay you can do, as I already showed you what kind of assay you can do to analyze the behavior or motor activity in the Drosophila. Advanced imaging techniques you can do, like you are expressing the particular protein in any particular tissue like neurons or eye tissue. Then by imaging you can identify, you can visualize the expression of that particular protein in tissue-specific manner clearly. So there are also development of the different therapies using AD models are in nowadays in treatment. Now with this, I would like to thanks to all for attention. I think I completed before time, because it was not the data and all these things, just an introduction of about Alzheimer's disease, how you can use the modeling Alzheimer's disease model. Thank you for attention.
Hello. Thank you, Dr. Singh, for informative lecture. Any questions? Anybody?
Thank you, sir. I'm also working on the Drosophila model.
Okay, good to know.
I'm working with the feeding model for Drosophila. We are using the Oregon R strain flies and we are inducing Parkinson's via paraquat and ecotoxicological induced model. The question I wanted to ask, sir, was when we give the oral dose of anything, either toxicant or the therapeutic agent, anything, when we give the oral dose, what mode will be better? Either should we mix it with the food and give to the Drosophila because the mouth parts are spongy type, and the next mode we use is the filter paper method. So which would you suggest is the better for absorption?
So the better method is always to give the supplementation in the food only, because the fly will be there in the food all the time. So whenever, because they will not continuously feeding on that food, but whenever they will feed on that food, they will take that chemical. So it will be better to mix your whatever the treatment you are giving or whatever compound you are giving in that food, so that they will keep feeding on that food. And one more important thing is you cannot measure the amount the Drosophila is feeding in that food. This is a kind of expectation that they are in that food and they are feeding. So they are taking that food. In that food, the chemical will also go inside the alimentary canal.
Yes, that was the point one of the reviewers raised, that whenever we feed them with the food, so we cannot measure that how much compound the one Drosophila has consumed.
That's why you are taking large number of data
Yes,
sets.
one data set we have about 60 to 75 flies
Yes.
for eating. But sir, they are asking that in the mice model they can tell that how much the dose
course.
they have given per kg of body
Yeah.
weight, like in LC50 and all those concentrations, they are able to mention. So they are saying that this is sort of a drawback for using the model, and they are asking.
No, LD50 here you can also count, because suppose
Yes,
you are
sir,
taking
definitely.
100 flies, if more than 50 flies are getting died by the supplementation, then you can monitor.
Yes,
But
sir.
single fly, you cannot say this single fly is feeding on that much compound and they are taking that much amount. You cannot determine. That's why you are taking large data set.
Yes, sir. They are preferring that we should use a filter paper model because then they are saying that at least the concentration is specific. In the food, when they are mixing, they are not able to consume the food.
No, even in filter paper, how you can say that whatever the compound you have given in the filter paper, they are consuming all the compounds.
So with the filter paper method, there is this contradiction that whenever we starve them for like 2 to 3 hours.
No, I am not aware, but for me, I always tested in the food only.
Okay.
So that is the best one.
उसका वापस बैठ कितना बचा है वो तो आप कैलकुलेट कर सकते हो लेफ्ट ओवर पार्ट को हम क्वांटिटेट कर सकते हैं और उससे पता कर सकते हैं पर इंडिविजुअल ड्रोसोफिला कितना ले रहा है ये नहीं पता
That is true. But what happened in like doses and all, you need to identify the lethal dose, what dose you are supplementing in the food. If that is called the toxicity of flies more than 50% or less than 50%, that dose we usually take as a consideration. We are not going to measure the dose concentration in the food left or Drosophila fly because this is a very small animal. So you cannot take individually, you cannot count how much in alimentary canal. Even you can do, if you extract the alimentary canal and you will go for MS and you go for others, then you can find it. But this is very difficult. This is not, and you cannot say accurately. It will be the amount which is uptaken by the fly and which is present in the. Sometimes excretion also happens. So maybe take it.
through mass up to femto level we can detect
Yeah.
equal to femto
Yeah.
level. Thank you, sir.
Any other question? Thank you, Dr. Singh.
Thank you so much.
We are reading about this model since decades. That is important for disease mechanisms, finding new targets and new therapies, etc. But most of us have not handled it, and even we haven't even seen how the Drosophila is used in lab or so. We were
Trust me,
having.
ma'am, this is very beautiful model. If you will work on it now, you will love it.
So
It's
see, there are only 2 people who have worked on this particular model and they are able to understand the
Yes.
things. But otherwise, Drosophila being
For
very
pharma
small
people-
creature, 0.3 millimeter, and will be alive for a few days, how the long-term studies are done, how anatomical interventions are seen, how the cancer studies seen, or even small things which we can easily see in the models, model, rabbit and all.
Yes,
And
yes.
for such
So
small
whatever
organism-
you can study in the mice model, you can study in the Drosophila also. But only the thing is that the lifespan is very small here in Drosophila. So you need to check the molecular level, not on the days or number of days you are experiencing the thing like in mice, you are in months you are experiencing, but their lifespan is about 60 days. Like and they will survive up to the 60 days. So the study, the Drosophila model is basically used to study the molecular aspects of any kind of disease, where you need to particular expression of protein in particular tissue, whether you are doing the histopathology by sectioning the brain, where the amyloid beta is expressing or not, tau protein is expressing or not. So that
means for doing long-term study also, or means like Alzheimer's disease is a progressive disease, it's a chronic disease, and
Right.
for such kind of disease, how this particular organism helps in finding the treatment options.
So treatment option is always you are using transgenic Drosophila, ma'am. You already expressing that you are not giving any chemical to induce the Alzheimer's in Drosophila. It is having that amyloid beta protein inserted in their genome already, and they start expressing from the larval stage. The protein will start expressing from the larval stage, and it will go up to the whatever the life expectancy of the fly will be. It will keep expressing and it will showing their effect. If you will check that effect in the neuronal tissue or eye tissue, that is up to you. Where you want to check the effect and in what way you are want to check the effect.
Thank you, sir.
Thank you, sir, for your magnificent talk. Now, I request Dr. Mukesh Kumar Meena and Dr. Vivek Jain to felicitate our speaker, Dr. Sandeep Kumar Singh. Now, I request them to felicitate chairperson and co-chairperson.
Now, before that, thank you so much, organizers of the conference, for inviting us for this conference.
I would request to Dr. Garima Joshi and Vivek Jain to felicitate chairperson and co-chairperson.
Nice.
Thank you, ma'am and sir. We have had a wonderful time so far. Now it's time to take a breath and refresh ourselves. I warmly invite you all to join us for a lunch in a cafeteria upstairs. We will reassemble after lunch for our next session. Thank you.